Hello everyone and welcome back to lecture 6, or the first part of it, lecture 6.1, where we will be talking about membranes. And in 6.1 we're going to kind of get ourselves situated for the rest of it by talking about these structures in the membranes, including various components and this concept called the fluid mosaic model, which is more or less how these components interact with each other. Now, before we get to that though, I'm sure you're all wondering, what happened with lecture question 5.2? Well, here it is. Which of the following are true about the structures in the endomembrane system? Now, if you said D, you'd be correct. Excellent. Normally, I want you guys to go and just figure this out on your own. And hopefully you do that before you get to these questions, right? Before you see the answers. That's kind of the ideal. But I'll kind of walk through this one real fast. So which of the following is true about the structure of the endomembrane system? Number one, Golgi modifies products from the ER. Well, we definitely know that's true, right? Remember the Golgi acts as the kind of shipping center, adding labels and stuff before it's moved on to where else, and somewhere else in the endomembrane system. Rough ER ships vesicles from the transface. Well, no. Number two, right? We didn't talk about a transface with the ER. That was with the Golgi. So number two's out. Number three, the mRNA is shipped from the nucleus to the ER via the vesicles. No, right? So the nucleus and the ER are kind of one of the few things that are actually continuous. They don't need that vesicle to ship stuff between them. So we can cross number three off the list. And then ER receives vesicles on the cis face. Again, doesn't have trans or cis faces. So 2, 3, 4, all wrong. Leaves us with one only, hence the answer D. Okay. I normally avoid doing that little exercise we just did there, where you can go through and eliminate them one by one. One, because it takes a bit of time. Two, I think it's at least as informative for you guys to actually go through that process yourself, right? If you didn't get D, that's okay. That's the point of these questions, is to help you identify what you're still struggling with. So it's up to you for most of the time to go through and say, hey, why aren't the other ones right? Can I go and figure it out based on my notes? If I can't do that, does maybe one of my friends know? That's also a great option. And if they don't know, does the forums? Maybe I can ask in the discussion forums on Sakai. Yeah, okay, that's kind of all I have to say about lecture question 5.2 and lecture questions in general for now. All right, let's move on to the content for today, where we will be introducing membrane structures. So, first off, if we're going to talk about membranes, we should probably, know what they're made of, the different parts. So what is the major structural component? Well, this slide should be almost entirely a review, since we've actually covered this lecture or this content like 3 lectures ago, and actually kind of again like 2 lectures ago when we talked about protocells. Speaking of which, figure 5.4, this lipid bilayer sphere, is actually a pretty good representation of a cross-section of what a protocell would look like. Oh, actually, we also talked about membranes last lecture too, with vesicles and all that stuff. So you should be able to answer all of this yourself, no problem, right? So what are the membranes made out of? No, don't cheat. Don't look at it yet. What are membranes mostly made out of? Right, phospholipids. Good. Okay, so what do we know about phospholipids? No, don't cheat. What do we know about phospholipids? Right. So they have a hydrophobic head and a hydrophilic tail. Good, yeah. What makes them hydrophobic? What makes the head hydrophobic? Yeah, of course, the phosphate group. Very good. How about the tail? What makes the tail hydrophobic? Did I flip that? I flipped that, didn't I? What makes the head hydrophilic? It's the phosphate. What makes the tail hydrophobic? It's the fatty acid, right? The hydrocarbon chain. Okay. Let's see here. We also have the glycerol that's also involved, and that's the glycerol itself is also hydrophobic. So the fatty acid and the glycerol are hydrophobic, and that phosphate group on the head is hydrophilic. Guys, you got all that. You got all those answers. That's great. It's almost better than me. I had to cheat a little bit. It's fine. Okay. So we know that's it, right? That's a membrane. Phospholipid automatically forms when you add it to water because the head is hydrophilic and the tail is hydrophobic, right? So we get this double layer. Cool, right? We're done. Now, there's more to it, right? So that is the main wall of this whole structure. The phospholipids compose a majority of it, but there's lots of other stuff kind of moving with and around those phospholipids. And next to it, actually, as well. So the most prominent are the membrane proteins, and these can do all kinds of stuff, often dealing with transport, getting things across the membrane or through the membrane or whatever. They can also move around the membrane, but they never do they flip, right? So it's not like they're usually oriented like in, here, right? So they go through like this, but they can't flip and go through because they have different parts of the protein structures are either polar or non-polar, which kind of determines where their orientation is and where position is. So, for example, you can be a integral protein, which they actually span the whole length or the whole width of the phospholipid, like this one right here. So this is an integral protein, goes from one side of the phospholipid bilayer to the other, and the kind of portions of that transmembrane protein is actually polar, and other portions are non-polar, which makes the whole thing amphipathic, right? So you have the non-polar portions that are kind of in the middle with the fatty acid tails kind of hanging out in the middle of the layer, and then you have the more polar sides of the protein hanging out on the outsides. Okay. Oh, you can also have peripheral proteins, which can be either polar or non-polar, and they kind of stick on the outside of the membrane. They can kind of even extend like a little bit into the membrane a bit here, as you can see, but they don't go all the way through. Okay. So what do these proteins do? Well, they have several functions. Well, okay, 6 to be more precise here, which are transporting things into and out of the cell through lots of different mechanisms, which we'll actually get to later on. They also can act as enzymes that facilitate reactions, and these are often taking place kind of right next to the plasma membrane. And these are, well, we'll get to that later. And then you can also have, as you can see here, they can function as part of signal transduction pathways, which basically means they help pass a message from outside the cell to the inside of the cell. We'll actually talk more on that later on in the course, I think mostly with, we'll get back to that. Anyway, so they also serve as, D down here, cell-to-cell recognition. In other words, kind of one cell being able to correctly identify with another cell. To be clear though, the cell-to-cell recognition is a product of a glycoprotein, not just a protein. So you have both a glycoprotein and on one cell and another kind of membrane protein on the other cell. We'll get more to that in a second here. And now remember what a glycoprotein is? What does this represent? Anyone? Bueller? Right, it's a carbohydrate, right? So how the cell-to-cell recognition works is that carbohydrate that's hanging off the side of the glycoprotein acts kind of like a key, and the shape of the protein on the kind of target cell is like a lock. So when you have the right key with the right lock, they interact with each other properly, and that's how you can recognize the cell you're looking for, right? Anyway, okay. Let's see, where were we? Now we're on to, E, intercellular joining. So you can have, let's see here, when a plant cells, like the cells of a plant are connected together, what's kind of holding those plant cells together? Some of these proteins, or heck, honestly, the proteins that keep your skin cells together, right? Finally, you can also have these kind of membrane proteins that are connected to the cytoskeleton and the extracellular matrix that you find inside eukaryotic cells. Now, this cytoskeleton and extracellular matrix, which we've maybe not talked about so much, it's okay. More or less what it does is it kind of provides structure in the cell for moving things around. Yeah, okay, so that's a lot of stuff we just talked about here. So there's transport, there's enzymatic activity, there's signal transduction, there's cell-to-cell recognition, intercellular joining, and then messing with the cytoskeleton and extracellular matrix. Whew. Okay. But, luckily for us, we really only want to focus on these first three: transport, enzymatic activities, and signal transduction. In fact, transport we're going to talk about in lecture 6.2, so the second half of lecture 6. Enzymatic activity, we're actually going to talk about a lot of proteins involved with enzymatic activity during the photosynthesis and cell respiration lectures, which are lecture 8 and 9 respectively. And then also signal transduction is going to come back to haunt you in Gen Bio 116 when we talk about cell-to-cell communication and hormones and stuff like that. So don't forget those. They'll definitely come back around. And I'm sure if you take like a cellular biology course, you're going to talk about all this other stuff too. But that's later on. That's a different course. Okay.